Hi, my name's Amy Holmes, and this is my Case Connection Unit 2 video. Today is 4/18/2020, and my two cases I chose were Case 2 and Case 4. In Case 2, the image shows a brain MRI of a large glioblastoma tumor with an area of necrosis within the tumor and a PET scan from the same individual's tumor following administration of fluorescently labeled D-glucose. The case connection asks, how does glucose uptake in normal tissue compare to glucose uptake by tumor cells in the MRI and PET image? And why would this be advantageous to the invasive tumor? The liver takes up and stores glucose to minimize fluctuations in glycemia in between the fed and starved states. The insulin pathway is the pathway in which the hormone insulin released in response to elevated nutrients in the bloodstream. Insulin triggers the uptake of glucose, fatty acids, and amino acids into the liver in the form of glycogen storage. In liver metabolism, glucose is taken up in an insulin-independent manner and enters through the pentose phosphate pathway, where it enters into the tricarboxylic acid cycle, which synthesizes cholesterol and fatty acids. Skeletal muscle metabolism, glucose is taken up through transporter GLUT4 and is insulin dependent, which means it only takes up glucose when insulin is present or in the fed state. Muscle will oxidize glucose through glycolysis, stores glucose as glycogen, and the TCA cycle will be active as a way to oxidize acetyl-CoA in an aerobic method. Adipose metabolizes glucose, is also insulin dependent, and metabolizes glucose through the GLUT4 transporter and oxidizes glucose by glycolysis. However, it will store glucose as fatty acids through the process of fatty acid synthesis and tag storage for synthesis. In the brain tissue, glucose is taken up in an insulin-independent manner through the transporter GLUT1 and GLUT3, and glycolysis occurring. Research has found that tumors will consume glucose at extremely high rates compared to the cells in which the tumor originated, despite the presence of oxygen. Glucose serves as a primary source for a variety of cellular functions: energy production, membrane synthesis, synthesis of nucleotides and lipids, and production of redox for antioxidative defense. Tumor cells gain specific enzyme regulatory mechanisms to direct the main fluctuation of glucose to those pathways. Cells located closest to the blood supply will present oxidative phosphorylation, as opposed to those cells farthest away will generate their ATP by anaerobic glycolysis. In the case of the two images of the tumor in the brain, the addition of fluorescently labeled D-glucose allows the patient's doctor to measure in vivo the rate at which the tumor is metabolizing glucose compared to the surrounding normal tissue to determine if a cancer treatment is effective in shrinking. Then the tumor will show less fluorescently labeled D-glucose in the image, as opposed to if there is no change in the tumor, then the fluorescently labeled D-glucose will show no change, or no growth or shrinkage. Research is looking at glucose utilization in cancer cells and the unique patterns of isoenzymes, which are the regulatory properties that allow tumor cells to develop their glucose-hungry phenotype. Case 4 presentation is a previously healthy 2-month-old with jittery spells several hours after breastfeeding. Laboratory results are consistent with hypoglycemia or low blood glucose, but otherwise unremarkable. Epinephrine was administered and there was no increase in blood glucose, but there was an increase in blood lactate. Physical exam revealed a liver edge 4 centimeters below the right costal margin. Percussions of the right chest and abdomen confirmed an enlarged liver. Blood glucose levels increased after breastfeeding, but the infant cannot maintain blood glucose levels within a normal range during fasting. After researching the infant's symptoms, the best diagnosis for this patient, I feel, is C, glycogen storage disease, or the inability to use glycogen. This disease presents around 3 to 4 months of age with an enlarged liver, elevated lactate and urate, hypoglycemic seizures, which is due to a defect resulting in an accumulation of glucose 6-phosphate that enters glycolysis, resulting in an increase in lactate production, causing lactic acidosis. The liver is unable to produce free glucose in response to neuroendocrine stimuli brought on by the hypoglycemia. A, hereditary fructose intolerance, is caused by deficiency of the enzyme aldolase B. Enzymatic block at aldolase B causes accumulation of fructose 1-phosphate. The accumulation has a downstream effect on glucogenesis and regeneration of adenosine triphosphate. After fruit juice and other foods high in fructose are introduced, infants would present with vomiting, convulsions, irritability, poor feeding, hypoglycemia, jaundice, hemorrhage, enlarged liver, hyperuricemia, and potentially kidney failure. This child, the mother reported that she had not fed the infant anything other than breast milk. Therefore, this would not be a correct diagnosis. Galactosemia is genetic disease, autosomal recessive, in which an enzyme needed to metabolize galactose is deficient or absent. Transformation of galactose to glucose is blocked, allowing galactose to increase to toxic levels in the body. Brain damage, jaundice, liver enlargement, cataracts, and susceptibility to infection. D, glucose 6-phosphate dehydrogenase deficiency, is a genetic disorder that occurs primarily in males and affects red blood cells. A defect in the enzyme glucose 6-phosphate dehydrogenase causes red blood cells to break down prematurely. This is called hemolysis, which leads to hemolytic anemia, or hemolytic anemia. Red blood cells are broken down faster than the body can replace them. Symptoms of this disorder are paleness, yellowing of the skin, and whites of the eyes, called jaundice, dark urine, fatigue, shortness of breath, and a rapid heart rate. This disorder is usually triggered by infection in the individual. Since the infant did not exhibit any of these symptoms, this would not fit as a diagnosis. This concludes my Unit 2 case connection.